opening ceremony and begin our scientific schedule. Now, we will have the introductory lecture of the Congress entitled Viewing Shallow Lakes from a Landscape Perspective, which will be given by Dr. Kendra Spence Cheruvelil from Michigan State University, USA. Finally, to present our plenary speakers today, to present Kendra, we invite Professor Dr. Ali Gher, member of the event's organizing committee. And before calling you, Ali, I would like to remember everybody that to watch Kendra's lecture, you just stay here in the same room. It will be in this room, the lecture, all the plenary lectures in this Zoom link, okay? Ali, the word is yours now.
Hello, everybody. Good morning, afternoon, night, wherever you are. Sorry for this crazy background. I'm trying to be official here, but I guess there's some interference. Anyways, so we now start with the first plenary lecture of our virtual conference, and it's a great pleasure and honor to introduce Dr. Kendra Cheruvelil. I hope I'm not destroying your pronunciation too much. So Dr. Kendra Cheruvelil is a professor in landscape limnology and co-director of the Data Intensive Landscape Limnology Laboratory at Michigan State University in the Department of Fisheries and Wildlife in USA. She has a dual PhD from the same institution in Fisheries and Wildlife and Ecology, Evolutionary Biology and Behavior from 2004. Kendra co-established the discipline of landscape limnology and conducts big data research on lakes to understand how global climate change and land use intensification affect lakes across regions and continents. She also pushes the boundaries of how to conduct limnology research by using data-intensive science, science of team science, and open science approaches, tools, and perspectives to answer complex scientific questions. After her lecture, there will be a 15-minute question and answer session. So please remember to use the Q&A tool in Zoom for posting your questions during her lecture. And without further ado, we'll now begin sharing her lecture. So let's see how this goes. I'm gonna stop my video now and share my screen. Can everybody see? Hello?
Yes, Ali, we can see your screen.
Okay.
Remember to put in a full screen, okay?
The volume isn't working. You need to unmute yourself, I think, to be able to, for people to be able to hear your computer.
Okay.
So you need to back it up.
There was a way to do this. We practiced this. I swear we practiced this. I'm gonna stop sharing and start again. Okay, let's try this again.
Good morning, everybody. I want to thank you for joining me this morning, and I want to thank the organizing committee for inviting me to share my landscape perspective and research with all of you today. I'm going to start us off with the basics of what is a landscape perspective. Now, often I think when people think about the landscape perspective, they are thinking about pictures such as what you see along the bottom of the slide here. Lakes within the landscape, embedded within a mosaic of trees, wetlands, cities, forests. And in fact, a landscape is any heterogeneous area made up of interacting ecosystems. However, when we think about landscapes, we're also very much used to thinking about maps. That's one way that we can delineate landscapes. And so here's a very common map that we're used to seeing. This is a map that shows you land use and land cover of the state where I live, in Michigan, in northern United States. And you see lots of green to represent forest, and then lots of yellowish color for agriculture and a little bit of urban. You don't see a lot of blue on that map though, which indicates wetland or open water, besides of course the Great Lakes surrounding the state of Michigan. Well, that's because a map is subjective. What you see on a map depends on what the cartographer decides to map. And so here's a different type of map where we could be thinking about the freshwaters of Michigan, and we could map the freshwaters. And now all of a sudden you see a map that's made up of a mixture of lakes in orange, wetlands in brown, and streams in blue. And now you have a very different perspective of the landscape. You have a perspective of the limnological landscape. And so when I think about how we want to apply a landscape perspective to the field of limnology, what I'm thinking about and talking about here is I'm talking about applying the characteristics and the principles and the theories of landscape ecology, things like patch characteristics, context, and connectivity, and spatial scale and the theory of hierarchy, and applying those to the freshwater landscape, being explicit about the roles of freshwaters, lakes, wetlands, and streams in the landscape, and thinking about things sometimes a little bit differently, such as when we think about connectivity and directionality, since water flows downhill and there are different water bodies that are connected to each other, we have to think very carefully about directionality when we're taking a landscape perspective of fresh waters. And so the idea is that we are explicitly including the terrestrial, the aquatic, and the atmospheric elements when we're thinking about limnology from a landscape perspective. And to do this, we really need information from local site-based studies of individual lakes, as well as broad-scale studies of populations of lakes. Now you might be wondering, well, what's the rationale for taking such a landscape perspective? Perhaps you've always studied lakes from more of a local or a site-based perspective, just like I did when I was getting my training. Well, one of the reasons that we advocate taking a landscape perspective is to really increase our scientific understanding of how lakes are going to respond to environmental changes, such as land use, climate, and atmospheric deposition. These sorts of pressures are broad scale. And so they don't happen on individual water bodies, and they can vary at large, say, regions and to continents. Another reason why we like to take a landscape perspective is that there are some gaps in scientific understanding that really can be best filled by such a perspective. So for example, we can only study a very small fraction of lakes. And so we have to do something called extrapolation, where we take knowledge from a few lakes and apply it to other lakes. We also need to scale up from a few sampled lakes in say one region or one continent to understand what's happening at the global scale. And finally, we need to be able to take understanding from the past and the current to be able to project and to forecast what's going to happen into the future. All three of these types of activities, scientific endeavors, are really helped by taking a landscape perspective. And that really includes the limnological or freshwater landscape, as well as the terrestrial and atmospheric landscapes. So how might we go about taking a landscape perspective? Well, I very much advocate that a landscape perspective is made up of, can be really advanced by using 3 types of sciences: data intensive science or big data science, open science, and team science. And so the idea here is that using open science perspectives and tools and team science perspectives and tools will allow us to really get new places scientifically using big data that we wouldn't be able to get otherwise, that there's real synergies by using team science, open science, and data intensive science. And so what I'm going to do with you today is talk to you about how I've applied those 3 sciences to using a filter of that landscape limnology framework to really understand lakes at a population level. And so first I'm going to share with you my teams, my science teams. And what I'm going to be, all the data that you'll see today, this is coming from these 2 amazing teams of people who I've been working with since 2011. We've got the first team of aquatic ecologists and GIS professionals, database experts and data scientists, started out at 3 institutions. And then in 2016, with our second grant, we got even bigger, added more statisticians, added some more quantitative based skills, and added another institution. And so we've got these 2 great teams working together to ask questions about lakes from a landscape perspective. And so the data that I'll show you today comes out from these teams, but also the lessons that we've learned is from working together as part of these teams for over a decade. All right, so again, how are we going to study lakes from this landscape perspective? What are the ways we can do that? Now, 10 to 20 years ago, this would have been a lot harder to do because the reality is that data looked like this. We had all sorts of data in hard copy, paper form, sitting in file drawers, all across the states of the US, all across the globe. And in fact, many scientists studied individual lakes one at a time, just like I did in my master's and my thesis. I studied 8 lakes, and then in my dissertation, I studied 54. However, my longtime collaborator, Pat Serrano, and I had this idea back in the early noughts that it would be important for limnologists to start thinking about lakes from this landscape perspective. And so our idea was to build a database with the needed information to better understand lake water quality using that landscape perspective. And so we embarked on this path of trying to build a database that would be research ready to be able to answer really important questions about lakes at landscape with a landscape perspective. And in essence, our idea was we wanted to go out and harvest all of those data that were sitting in individual file cabinets across the Northeast and Midwestern US and bring it all together and harmonize it and come up with a research-ready database for everybody to use. And so we started scouring for data. We have lots of different data sources out there. We went to state and tribal natural resource agencies, to federal agencies. We went to what's called citizen science programs, volunteer programs coordinated across communities. And we reached out to individual university scientists who have big lake programs. And again, we were really focused on how do we build on how do we take what is useful from open science and team science to be able to do the best possible data intensive science. And so this is where Lagos was born, at the intersection of these sciences. And so our first phase of research, the first team that you saw on the top of the team slide a few slides ago, was to build Lagos Northeast. So the Lake Multiscale Geospatial and Temporal Database is called Lagos. And we started with the Northeastern and Midwestern US, where there are about 51,000 lakes greater than 4 hectares in a very large area of the United States. And so you see those here. You see the states listed in this inset box, and then the red lines here are what we call HUC4 regions. And the blue dots obviously are the many lakes in that area. And here are examples of the lakes for which we have long-term data within our Lagos Northeast data set. So the Lagos Northeast data set is both spatially extensive and temporally extensive. So we have, there's 51,000 lakes in the database, and for about 20% of those, we have some in situ data. And then for a smaller percentage of those, we also have data through time. Isn't that great? So who sampled these lakes? Where did they come from? I listed all these different places that we went to try to compile data for Lagos Northeast. When we did an analysis of our resulting database, what we actually found was that a vast majority of our data are collected by non-professionally trained as scientists samplers. So as it turns out, we have citizen scientists collecting a vast majority of the data. Here on the left is water clarity measured by Secchi depth. As you see, through time, Secchi depth has always been something that people have gone out to their individual lakes and sampled and reported as part of coordinated programs. Around 75% of our Secchi data comes from these sorts of programs. On the right here, we have phosphorus, which of course is a much lower number of overall samples than water clarity. And if you look between 1980 and 1990, most of the phosphorus data was coming from other places, such as state and national agencies. However, through time, and when you get out to the last decade, almost 75% of phosphorus data is also coming from citizen science or coordinated volunteer monitoring programs. So we would be in a sorry place if we didn't have these amazing lake property owners going out and sampling and reporting the quality of their lakes. The same goes when you look at that sampling through time in terms of where do we have long-term data records. And so again, if we look at through time, how many years of data now we have sampled, again, the lakes for which we have long-term data, so at least 15 years, it's almost exclusively from citizen scientists for Secchi depth and almost 75% of phosphorus data. So we are so very thankful to these community organizations that have been collecting water quality data all these years for us to be able to use to ask and answer really important questions about lakes. So here are some lessons that we learned by building Lagos Northeast. First, we need to study lakes both individually and at the population level to continue to understand, manage, and enjoy lakes. Second, we can compile existing data to study lakes at the population level. Now, this is no easy feat. It took years and lots of money to build Lagos Northeast, but it is possible. Three, citizens collect a lot of data, and that data make up a high proportion of what is available to us as scientists and for managers to really understand what's going on with lakes using a landscape perspective and through time, at least for the Midwestern and Northeastern US. So then we started to dig into Lagos Northeast in terms of how healthy are the lakes in this part of the United States. And so here I show you a violin plot that shows you chlorophyll A, so an indication of trophic state. And you see up at the top, these are the lakes that are more eutrophic, mesotrophic, and at the bottom, oligotrophic. And generally what you see in Lagos Northeast is that lakes are actually really pretty healthy, that the vast majority of lakes are either mesotrophic or oligotrophic. And then yes, there are some eutrophic lakes up there at the top, but it doesn't make up the majority of lakes in the Midwest and Northeastern US. You might also ask, how big are sampled lakes? Well, we looked at this by state. So here you have the US state, which for some of you won't mean too much. But the idea here is that area on the x-axis here, and I give some comparisons here from Canada, Switzerland, and Guatemala in terms of size. What you see is that if we look at all lakes in the Midwest and Northeastern US using our census lake population, lakes are relatively small. Looking at these stars, somewhere between 25 and 100 acres. However, if you look at the sampled lakes, they are much bigger, right? And so what that's showing is that there is a bias towards sampling larger lakes in these Midwest and Northeast United States. What type of lakes are sampled? We have a variety of lakes, types of lakes based on their surface water connections. So for example, in Lagos Northeast, there are lots of isolated lakes with no inlets or outlets, headwater lakes with just outlets, run-of-the-river connected lakes, which we break into two groups, drainage stream, where they have an upstream, just streams, in and out of the lake, and then what we call drainage lake stream. And these are lakes that are connected both to streams and to upstream lakes. And so what we wanted to know is, are there biases in how these are being sampled? So we saw in the last slide that we tend to oversample large lakes. How about different types of lakes based on connectivity? And so first I'm going to show you what proportion of lakes are each of these categories of connectivity. All right, and so basically what we've got is we've got about 30% of lakes that are have streams upstream and downstream. About half of lakes almost are completely isolated in the Northeast and Midwest US. 15% are headwater lakes, and then about 10% are the really connected with both lakes and streams upstream. Now, how many of these, or what proportion of these are actually sampled? So where do we have their water quality data? So headwater lakes and drainage stream lakes, we actually find are sampled just about proportionally to how often you find them in the landscape. Where we see a mismatch is with the isolated and the drainage lake stream lakes. So what we see is that the highly connected lakes that are connected to streams and upstream lakes are oversampled, and the very disconnected and isolated lakes are highly undersampled. And so this just shows you again, this idea that our sampling that we are conducting, not coordinated across any program, but all of the sampling that's happening by different agencies, by citizens. When you compile all those data, you end up with data that have fewer lakes sampled that are isolated and more that are very, very connected. So what lessons have we learned from this part of the talk? Lakes sampled in the Midwest and Northeastern US are generally healthy, large, and connected to other lakes, especially when you compare that to the overall lake population, the census population. Okay, so this is a shallow lakes conference. You might be wondering, when are we going to talk about shallow lakes? So here it comes. Here's our case study about shallow lakes. I'm going to share with you results from a project I've been working on with Ty Wagner and Kath Webster and Caitlin King and Autumn Poisson, where we've been looking at the shallow lakes in Lagos Northeast and trying to understand what are the patterns and drivers of total phosphorus and chlorophyll. So I'll start with just a little bit of background. Obviously, I'm very keen on taking a landscape limnology perspective. And it's true that over the past 2 decades, as landscape limnology has become more common, we've learned lots of cool things about limnological relationships, such as the positive relationship between Tp and chlorophyll. The fact that those relationships can vary regionally, and so you see patterns, differences across regions in those relationships. That those relationships can be driven by multi-scaled ecological context drivers. So things like a watershed, percent of watershed that's urban can drive that relationship at the same time as the depth of a lake and the regional agriculture in the area. And finally, we've seen results that show how important lake connectivity is for trying to understand broad-scale patterns in lake water quality. Now, at the same time that all this research has been happening, we of course also have lots of seminal shallow lake research that has led us to all sorts of really great ecological theories and management strategies, such as alternate stable states and biomanipulation and regime shifts. However, there has been, until relatively recently, some gaps in our landscape scale shallow lake understanding. And so what we wanted to do was we really wanted to look at what are the landscape patterns of states and relationships of shallow lakes in Lagos Northeast, focusing on total phosphorus, chlorophyll, and then the relationship between total phosphorus and chlorophyll. And we wanted to look at what were the multi-scale drivers and interactions that were related to those patterns. Now, today, what I'll show you is on focus simply on two of those variables, connectivity and regions. And of course, this is the perfect place to be talking about this kind of work, because you're going to be able to go to so many sessions this week that have this same idea, this landscape lens that people are using into their research, such as this session 2 about the role of connectivity in the structure and function of shallow lakes. That is was put together by Linda and Lawrence. And session 12, linking functional traits with ecosystem function across landscape scales by Ali and Miriam. And so lots of great talks that are taking the same landscape perspective. And I will just plug one talk by Laura Sanchez, who came and spent some time in my lab last year, who's going to be talking about the Pampian shallow lakes taking a regional approach. Okay, so you have seen many maps now about Lagos Northeast, showing you all of the lakes, 51,000 lakes, and showing you the lakes for which we have in situ water quality data. Well, now let's think about how many of those lakes do we have lake depth information for. It turns out that of the lakes in the Lagos Northeast study extent, only about 16% of them have lake depth data, but that's still 21,000 plus lakes, right? Out of the 150,000 lakes in that area, we've got 21,000 with lake depth. And so we wanted to subset the data and really focus in on shallow lakes. Now, we use depth cutoffs of a mean depth of less than 3 meters or a max depth of less than 5 meters if we didn't have mean depth for a lake. And what you see here is a map of shallow and deep lakes in this Lagos Northeast study extent, where shallow lakes are in blue. And it turns out that shallow lakes, for the lakes that we have depth data, shallow lakes account for 44% of those lakes. And they're very nicely distributed across our study extent. And they're, both big and small. Here's a really big one, right? And so we were really excited by this. That's a huge number of lakes to be able to start playing with data on. However, this is just the lakes in the Lagos Northeast study extent that have depth data. Then we had to figure out, well, how many of them have TP data, how many of them have chlorophyll data, and how many of them have both. So our sample size doesn't stay, unfortunately, at this high of a number. So first we asked, how connected are shallow lakes? These are the same 4 categories that I showed you a few slides ago. Now I am going to combine isolated and headwater lakes in this analysis. And so what you'll see is 3 categories: drainage lakes that have upstream streams, drainage lake streams that have both upstream streams and upstream lakes, and then more isolated lakes that have no connections whatsoever or only have outgoing streams. And here is a figure that shows you those 3 connectivity classes and the distribution of shallow lakes by those classes. And so what you see is that generally they're pretty evenly distributed in terms of the proportions across these 3 classes are about equal. There's about 30% of each type. There's also some good spatial representation across the three groups, although you'll notice, right, that isolated and headwater lakes are much less common down in the southern tier than they are up in the northern tier. So then we wanted to look at, okay, so is there a regional pattern to TP and chlorophyll? And so again, we used what we call HUC4 regions for our study extent. We selected out total phosphorus and chlorophyll data from the summertime using June 15th to September 15th, epiliminetic concentrations between 2002 and 2011. And so that led us to just over 2,500 lakes. And what you see here is just maps of median total phosphorus on the left and chlorophyll on the right across these HUC4s. And a couple things just to point out, right? So dark blue is oligotrophic, right? And it's interesting, by chlorophyll, you see that the northern tier here, they're on average, those regions are oligotrophic. However, you don't see that if you use TP as for the cutoff for trophic state. But again, nice distribution. Another thing you'll notice is that most of our hypereutrophic lakes, whether you look at TP or chlorophyll, are in the southwestern part of our study extent. That area is part of our very high agricultural part of the United States. It's part of the breadbasket or the corn basket, depending on what you want to call it. But there's a lot of agriculture in that area, very flat, very fertile land. So that makes sense. But overall, regional patterns, right? High water quality leading down south to west to lower water quality. So how about that relationship between TP and chlorophyll, right? We know in general there's a positive relationship. And in fact, some previous research that has looked at taken a landscape perspective to the TP chlorophyll relationship has actually found nonlinear threshold effects that occur between total phosphorus and chlorophyll. We did not find that in our sample lakes. So in fact, for shallow lakes in northeastern and midwestern US, we found relatively linear relationships between TP and chlorophyll, if there was a significant relationship at all. How about the role of connectivity for understanding broad scale patterns in these two variables? So here, what I'm showing you are box plots, and we have connectivity along the y-axis. So from most isolated to connected to upstream streams to connected to upstream streams and lakes. And so at the top, we have total phosphorus, and below that, we have chlorophyll. And I'll just point out here, right? So here are the cutoffs for oligotrophic, green, meso to eutrophic, and that orange, yellowish color for eutrophic. And again, pointing out that what's interesting here is that connectivity matters in terms of the more isolated the lakes are, the lower the total phosphorus and chlorophyll happens to be, okay? And so the more isolated shallow lakes tend to be more oligotrophic than the connected ones. Well, when you look at that relationship between total phosphorus and chlorophyll, we see that still that positive linear relationship, and we see no effect of connectivity class on that. So there's no difference in slopes between isolated lakes and connected lakes and very connected lakes. So what did we learn by looking at shallow lakes from this landscape perspective using Lagos Northeast? We saw regional patterns in total phosphorus and chlorophyll, with that relationship between the two being relatively linear both within and across regions. And we saw that connectivity mattered for states with more isolated lakes having lower TP and chlorophyll, but that it didn't seem to matter for the relationship between TP and chlorophyll. But overall, one of the main one of the main take-home messages for us doing this research was that there are a lot of shallow lakes in the northeastern and midwestern US, which has huge implications for scaling, estimates of global processes, all sorts of really important questions facing limnologists today. However, we are a little limited in our ability to really understand what's happening with these shallow lakes and their roles in these global cycles because of our lack of depth data. Remember that we only had depth data for about 16% of the lakes in our study extent. So using the number of 44% of lakes with depth data being shallow, we can estimate that there's probably about 66,000 shallow lakes greater than 1 hectare in surface area in this study extent. So lots and lots of lakes out there for us to be able to learn about if we had the depth data. So connectivity and or region are important for understanding patterns of total phosphorus chlorophyll and total phosphorus to chlorophyll in shallow lakes when you're taking that landscape perspective. And we really should be thinking about explicitly about studying shallow lakes from that landscape perspective. All right, so far I've been sharing with you results from phase 1 of our research, which was focused on building Lagos Northeast, which you see here. Phase 2 of our research, I tell you, we are a glutton for punishment. We decided at the end of our first project that we wanted to go bigger. We wanted to create a conterminous US database that would be available for researchers. And so we are currently working to finish up Lagos US. And so you see here the patterns of lakes across the US, really amazingly rich areas of across the US. And in fact, our Lagos US data platform is nearing completion. And so right now we have 6 modules that are all in the works. At the center is Lagos US Locus. And that one is under review and will be out shortly in L&O letters. And that provides all of the census information. So all of the lakes in the United States, their watersheds, their connectivity classes, their names, all of that sorts of locational information. And then we've got 2 of the 3 modules down here. One is the networks, so connectivity networks, the quantitative measures of connectivity. That extension module is also under review at L&O letters. And we've got Lagos US Reservoir that I'm going to show you a little bit about today too, which is about to go in. And so we've got these modules that we are releasing to the public shortly and will be available for everyone to use. So let me share a little bit of information about Lagos US. So this is what we've learned so far. What are the most common US lake names? So if I had a poll, I would give you a poll and have you answer this question and see what you came up with. And it's kind of a trick because the most common US lake name is no name. So in the US, there's a formal naming system. And in that naming system, 83% of lakes have no name. So it's a little bit of a trick. But if you look at the 17% of lakes that do have names, the most common lake name, yeah, it's mud. We are so very creative in our naming, aren't we? And we also have found that there are some regional patterns in how humans have named lakes through the years. And so this map is showing you that along the East Coast, we tend to call things mud pond, whereas in the center of the US, we call it mud lake. And then in the western part of the US, we call it mud reservoir. And at the very southwest, it might be called mud tank. So lots of really cool things you can do with a database of all named lakes in the US, showing our creativity or lack thereof and patterns in how we name lakes. We've also asked how much and where does connectivity fluctuate across the US? And so using these same 4 connectivity classes that you've seen twice now, we mapped where changes in that connectivity happens. So this is based on the stream systems that are ephemeral. And what you see here is that in the middle US especially, and in the southeast here, we get lots of fluctuations in connectivity. And so that's showing us that connectivity is actually quite dynamic, especially in Middle America. We've asked questions about how many and where are lakes being protected. And so here is a map showing you across the US the percent of a lake's watershed that is protected. And so the dark blue here is what you're looking for, is most of the watershed is being protected. And what we see is that only about 10% of lakes have most of their watershed protected. And that most of those lakes, as a high proportion of those lakes, are in mountainous Western US. So interesting patterns in how lakes are being protected and where. We've also asked how fire might be affecting lakes. We often see articles in online news bulletins and news sources showing dramatic pictures like this of fires in the Western US affecting lakes. But it isn't very common for limnologists to study the effects of fire on lakes using a landscape perspective. And so we tried to do that. And what we found was that there are a lot of lake watersheds across the US that have high percent burn in their watersheds. And so the bigger the circle on this map, that's showing you the more of the watershed that has been burned through time. And so you see, yes, a lot of that is happening in the Western US, which we hear about in the news all the time. But there's also a lot in Florida, along the coast, and in the northern tier of Minnesota and Wisconsin. And then we also looked at how that's changing through time. And so here on the x-axis, you see year. And what you see is that the prevalence of fires has been increasing, especially since the early noughts. And a lot of that is wildfire, that's the red. And so fire is increasingly affecting US lakes and their watersheds. And so this is a really important area that needs research on lakes using a landscape perspective. We've also asked how deep are lakes in Lagos, US. So that case study about shallow lakes that I showed you was only for the northeastern and midwestern US. Well, what about if we apply that to all lakes in the US? And so we have an extension module for Lagos US called Lake Depth that will include all of the lake depth data that we were able to scour from any data source. And that is in preparation. And what our graduate student Joe found was that there's a dearth of lake depth information out there. And in fact, there's only about 4% of lakes that are with a minimum cutoff of 1 hectare in size that have depth data available for them. And of course, this is a problem because it means that we can't answer the question of how deep are lakes in Lagos, US, but it also is a big problem because we know lake depth is so very important for many in-lake processes. And then that of course affects water quality and being able to have safe drinking water and recreation and so on and so forth. So again, really a dearth of depth information that we need to deal with. We've asked how many lakes are reservoirs. So in Lagos Northeast, when I showed you the map of all of the lakes in the Northeast, I was including lakes and reservoirs. We had no way to differentiate between the two. Now for Lagos US, we really wanted to be able to classify lakes as either natural lakes or reservoirs. And so we have built an extension module to Lagos US that is a classification of whether each water body greater than or equal to 4 hectares is either a natural lake or a reservoir. Now, we did this using a machine learning modeling approach, and that's because reservoirs are very hard to differentiate. Here's just 2 examples of aerial images that we used for training our model. And you can see that reservoirs have a variety of shapes, and we certainly could not look at an aerial photo for every 150,000 lake, all of the 150,000 lakes over 4 hectares. So we built a model and we define our reservoir as a lake that is likely to be either human-made or highly human-altered by the presence of a relatively large water control structure that significantly changes the flow of water. And this was based, again, on visual images, images like these satellite images that were used to train a model. And so this is based on aerial imagery and being able to visually detect either a dam or a water control structure that was pretty big. And guess what? We were then able to apply that to all of the lakes in Lagos, US. And here what you're seeing is where and how many of the lakes that are greater than 4 hectares are reservoirs. And what you see is that 44% of lakes are actually reservoirs, so human-made or with a substantial water control structure. And you also see that they are throughout the US. There has been in the past, I think, a misconception that reservoirs are really something that you only find in the South, but actually reservoirs are very common in the northern tier of the US as well. And then we asked, well, okay, so now we know which ones are reservoirs. How many of those might be shallow reservoirs, right? And so this idea of we want to understand shallow lakes, well, that means all lakes. When we say shallow lakes, that includes both natural lakes and reservoirs. So we used our reservoir database, and we had depth data for about 6% of reservoirs. And here what you see are the reservoirs and natural lakes. So reservoirs are in the pink. Across the US, right? And so we see the reservoirs again are all across. So these are all of the ones that have depth data. And 44% of those reservoirs that had depth data were shallow. So very interesting. This 44% is seems to be very consistent, whether you're talking about natural lakes or reservoirs, that about 44% of lakes are shallow. And so if we take that percentage and we estimate out to all of the US for the lakes and reservoirs for which we don't have depth data, that means we likely have about 27,000 shallow reservoirs across the conterminous US. Again, a huge number of shallow systems that we could be studying. So the lessons that we've learned from just the beginning of using Lagos US is that there's just so much to learn about lakes from studying them with a landscape perspective. And that's all lakes, shallow or deep. Examples that I gave you is, broad scale patterns in lake names, connectivity, fires, prevalence of reservoirs, but I mean, the list could go on and on and on. And hopefully I've piqued your interest if you don't already, at studying lakes or shallow lakes from a landscape perspective. And if so, you too can use Lagos Northeast to do so. So Lagos Northeast is completely available to everybody. It is published on the Environmental Data Initiative data portal, freely available there. We have extensive metadata. We have a data paper that describes Lagos Northeast, and we have an R package available for easy access and manipulation of the data. And soon, sometime this spring or summer, the first phases of Lagos US for the entire conterminous US will also be available publicly to everybody anywhere to use. We are very committed to open science perspectives and tools. We would not have been able to compile these data and make these databases if it weren't for all the people sharing their own data with us. And so we make our data products and data available to everybody as well. So look for those coming soon, and I hope you all join me in viewing lakes from a landscape perspective. Thank you.
Hello, everybody. I hope that went fine. Did everybody hear it well? Great. So that was a really cool presentation, Kendra. I especially like seeing how the evolution of the the monitoring sites over time, like you said, it's really cool and powerful to see. Hopefully we'll be able to have a Lagos world sometime in my lifetime. That would be nice. So right now I'm gonna ask you to turn on your microphone, and you're already sharing your face, so that's nice. So I'm gonna also open here the question and answer session. I wasn't able to follow the chat, so I don't know if people ask their questions in the right place. Can somebody from the organizing committee tell me if there's questions in the chat?
Lee, there is one question from Marianne. You
Yes.
can read, and I would like to remind people to forward their questions through the question and answer and leave the chat for other conversations, okay? And I will also, I have just sent now through the chat the link where Kendra's lecture can be available in case you want to watch it again or share with some colleagues.
Okay, thanks, Koka. So I'll open the first question, and it's from Mariam Bekliolu. Mariam says, Kendra, thanks for a great talk. How about biological data which are costly to get, but there is a program maybe for the subset of them, since macrophytes and zooplankton can be crucial to understand all these interactions, especially in shallow lakes?
Yeah, that's a great question, Miriam. We have for years and years wanted to have a biological module for Lagos Northeast and then for Lagos US. Obviously, many of us are really interested in responses by biota to multiple stressors. As you might imagine, trying to compile biological data from multiple sources is very complicated. We don't have quite the same standard protocols in place for processing of biota as we do for things like nutrients and chlorophyll. So it makes it pretty difficult to do that. However, we've made some progress. Obviously, we have chlorophyll in there as a proxy, as a measure of productivity. And this in Lagos US, we'll also have microcystin, so we'll have some indication of toxicity from microcystis. And then we have different people in our team who have been working to compile biological data to complement Lagos. And so it's not technically part of Lagos Northeast or Lagos US, but it is being used in concert with those with our data products. So for example, my graduate student, Caitlin King, has worked with the US EPA National Aquatic Resources Survey data. So that's the NARS survey data, which includes macrophyte measures and zooplankton measures. And so she published a paper, I think it came out in 19, that used those data. And some of those data are still included we pulled into Lagos. And then Caitlin King, she went on to study, she combined data from streams and lakes about fish species diversity, and then has combined it with Lagos Northeast data. And so she's got a paper that's coming out, it's in press with the Canadian Journal of Fisheries and Aquatic Sciences, so it should be out soon. That's looking at patterns and drivers of fish species composition so from a landscape perspective. And you know, that took a lot just to get 5 states worth of fish data pulled together. So it's a big endeavor. And then we've got a couple of graduate students in our lab who are working on linking Lagos to USGS and Sea Grant data about zebra mussels and quagga mussels, and then to US Fish and Wildlife data about breeding waterfowl. So those are some sort of efforts that we have ongoing to link with biota. I agree, it would be wonderful to keep building up the biota in our database, and it's something that we really are interested in doing. It's very, very difficult. But thanks for the question.
So onwards, according to the order of the questions we received, the next question is from Sari and Kostin. And she also thanks you, Kendra, and she wonders how you dealt with quality control of the data. And I think many people have similar questions.
Yeah, quality control is a big challenge that we tried to tackle head on. So there's a few things we did. Each data source, when we pulled data from government agencies, citizen science programs, wherever we pulled it from, we asked for their detailed methods, both field and laboratory methods, and we authored just hundreds of pages of metadata to accompany each of those, all of those different data sources. And so we went through the methods used. And so that was sort of on the front end. And though we have published very detailed user guides, and then the metadata are embedded, you can link to metadata as well in with our database. But then after compiling all the data, we also did some sort of QA/QC on the back end in terms of looking for anomalous values, we would do plots where we would look at relationships, known limnological relationships, and look for really weird values. And what we did was we did not delete any of those what we thought were anomalous values, but we flagged them. So we have a very complicated system. Well, I guess not complicated. We have a lot of flags in Lagos Northeast and Lagos US where users can go through and they can make their own decision about whether they want to include particular observations in their analyses based on the sets of flags that we came up with. And so we did flagging for all sorts of different reasons, and we describe all those in the user guide. And so that was sort of that's the basics of how we went through and dealt with quality control. Was on the front end, making sure that we had all of the methods and that we documented all those methods very carefully through metadata creation. And then on the back end, doing a lot of both univariate and bivariate, but then some multivariate using machine learning approaches, some sort of plotting and visualization and looking for values that needed to have flags inserted.
Okay. And the next question is from the Saras Institute. They start out by a joke. They say, what's a shallow lake? A lake where the fish are crouching. A definition from Argentina. But then they continue, seriously speaking, is it possible to deepen the rigorous identification of shallow lakes due to the absence of prolonged thermal stratification?
Oh, that's really interesting. So in Lagos Northeast, when we started with the Northeast and Midwest US, we did not collect, well, I guess we did collect, but we did not publish temperature data. However, Lagos US that we're working on now, we did actually collect, and we're working on a profile module such that people could go through and look at temperature and oxygen in lakes. Now, not every lake will have profile data available, but certainly that could be used as another way to identify shallow lakes. So that's a really great idea, and I'd love to see how that played out. When we've been playing with the data and looking at questions related to shallow lakes, it is really amazing how you look through the literature and different depth cutoffs have been used around the world, depending on timing and where you are. And as I said, we don't have data for a huge proportion of lakes in terms of, we don't have depth data. Now, the lakes that have thermal profiles are likely ones which we do have depth data, so we still won't be getting anything new about lakes without depth data. But yeah, those data could be used to do that, and that would be really, really cool.
The next question is from Tom Nijman. Sorry for pronunciation errors there. Thank you, Kendra. Most of the data you showed in terms of eutrophication was about phosphorus. Do you have any idea about the patterns in terms of nitrogen?
We do. And I haven't looked at it recently, so I'm not sure I can speak very well off the top of my head. But we obviously have total nitrogen data as well, and we've plotted those. The issue is that the amount of data does drop quite a bit. So for example, we have the most data for variables that are quite easy to measure, like Secchi depth or water clarity. You know, if we think about an area where we might have 20,000 records of water clarity measurements, then we usually have about a third of those we will have total phosphorus data for. So we might have 8,000 records of phosphorus data. And then another half of those we might have total nitrogen data for. And so the reason I chose to share the total phosphorus and chlorophyll data is because we have so much more of it than we do for nitrogen. But we do have those data, and we've had some people in the group have done a really cool work looking at Tp, Tn, and chlorophyll, and looking for threshold effects and what might be driving different relationships. And some parts of our, so in the agricultural area, for example, total nitrogen can actually be more of a driver than total phosphorus. So we definitely have been looking at those sorts of things.
Next question from Lawrence Carvalho. And he says, great talk, Kendra. The slopes of chlorophyll TP ratio differ geographically, and looks like median TP and chlorophyll differ regionally in the northeastern US. What do you think controls these regional differences in chlorophyll compared with TP? For example, in southwest of HU4, I already forgot what that was.
Yeah, I know what he means. Yeah, hi Lawrence. Well, we have in the US, right? It's huge, and there's a very wide gradient of soil types and you know, glaciation history, and so naturally in the US, when you move from the Northeast and you move south and west, we end up with areas that are much more fertile soil and naturally have higher productivity lakes. That happens to, of course, then be where our agricultural region of the country is. And so then you end up with even more cultural eutrophication as big farms are putting lots of nutrients on their fields. So it's both a natural phenomenon, and then also it's increased by fertilizers.
The next question is, the names are getting more exciting. Dianeke, I won't even try the last name. Thank you for the interesting talk, Kendra. Very nice to hear more about Lagos. I wonder whether you also explored whether the lower TP and chlorophyll levels for isolated lakes versus connected lakes could be explained by the connectivity or other environmental factors that might be reflected in the regional pattern of lake types.
Yeah, great question. Yes, we have looked into that some. There obviously, when you're thinking about these sorts of things, there are some I don't know, confounding factors, for we could call it, but it's just, you know, varying patterns. So for example, it could be, you might think, well, maybe isolated lakes happen to be in areas that have lower urbanization, for example, and that might be partly why you see those patterns. And that is true in some cases, but if you think back, the slide that I showed where there was a map of the different lakes by connectivity type, those isolated lakes are actually spread regionally. They're all throughout the study extent. They are not just, so for example, they're not just in the northern tier where there isn't as much urbanization or agriculture and more pristine areas. Those isolated lakes are all throughout. And so there may be a piece of it that's about where they're setting, but it's not simply that.
Next question is from Meredith Holgerson.
Mhm.
And she says, excellent talk and such exciting new work regarding Lagos US. I'm curious about the size threshold. Mostly you discuss lakes larger than 4 hectares, but occasionally mentioned 1 hectare. Is there an effort to get these smaller water bodies included? My guess is the number of shallow systems is much greater than 44% if you reduce that size threshold.
Yeah, great question, Meredith. Thank you for asking about that. So when we built Lagos Northeast back in 2011, we used 4 hectares as a minimum size cutoff for a lot of our work, in part because of analyses that we did with our data that came from our landscaping data that came from satellite images and looking at where error rates got big, and it was between that 1 and 4 hectares. We were unable, the resolution wasn't good enough. And so that's why the 4 hectares was the cutoff for Lagos Northeast. However, in the last decade, things have gotten better. And so for Lagos US, our minimum lake size is actually 1 hectare. And so when all those data are released, you will see that the minimum lake size is 1 hectare. And I agree, we would think that the smaller size water bodies, that a higher proportion of those may very well be shallow. So it could be that it will be higher than 44%. I did also want to mention, so the 4 hectare cutoff, we still kept with that cutoff for our reservoir modules. So Lagos US has all these different modules. And one of the extension modules that I talked about at the end was reservoirs. And so that classification of whether lakes are natural lakes or reservoirs. And we did need to keep that 4 hectare cutoff for that module, unfortunately, because that was built on a machine learning model that used satellite images to identify dams and to visually identify water control structures. And we just couldn't, there was too much error when we went down to 1 hectare. So although most of Lagos US will go down to 1 hectare, the reservoir module still has to cut off at 4.
And there's a related question that maybe was already answered right now from David Cunilera Motcusi. Thank you, Kendra, for this nice talk. How about small-sized water bodies, smaller than 4 hectares, that we could call ponds or small lakes? Did you try to get information regarding these systems? Probably more abundant than the others. So,
yeah, the numbers are staggering. When you go from 4 hectares as a cutoff down to 1 hectare, the numbers of lakes are it's just amazing. And so we'll be able to do all sorts of really fun analyses with Lagos US, comparing what we know based on using a 4 hectare cutoff with a 1 hectare cutoff. And so I'm really looking forward to that. Now we do know, unfortunately, that our sampling programs are biased toward larger lakes. And so even though we'll have numbers and we'll have landscape and climatic variables and watersheds for those smaller lakes, the proportion of those smaller lakes for which we have in situ sampled data is likely going to be relatively small, because state agencies and university researchers and so on and so forth, we have a bias towards sampling larger lakes.
the next question is from Mariana Mirhoff, and she says, hi, Kendra, great talk. In connection to Lawrence's question, besides, oh, wait, it's moving as I'm speaking. Besides land use, such differences in Tp chlorophyll slopes couldn't be the result of different trophic interaction. For example, lack of grazers removing algal biomass due to various reasons.
Yeah, definitely. And that's one of the reasons that one of my grad students is pulling in information about zebra mussels and quagga mussels, right? So for example, they clear water. So even waters that we would generally think would be high in chlorophyll, if they have zebra and quagga mussels in those systems, it's going to be very different. And so some of that could be at play. And so yeah, that is one of the really cool things about having biological data, is to be able to get at trophic interactions, which we haven't been able to do thus far very much.
Yeah, that would be great. So I'm I don't know what's happening with the order of the questions. I'm just going to scroll to the top. So sorry if I'm pulling over anybody here, but anyways, the next question is from Sumita. And thank you for an in-depth presentation. Is flow data also being included into the data set? How many lakes would have such flow or volume data? And if this could be used for preparing management plans for TM, total maximum daily loads for lakes in different regions of the country?
Yeah, that's a great question. So flow data would be for the subset of lakes that are connected, obviously, to rivers. And so we would think about that would be about 50, yeah, about 60% of the lakes would be connected to upstream rivers and or lakes. And so it would be a subset of the lakes. We don't have flow data in Lagos at this point. There are gauging stations that different agencies put in rivers and streams, like the US Geological Services, they do that. We haven't thought about trying to incorporate that sort of data into Lagos, and I'm not sure that's a really good thing to be thinking about. We don't have that at this point. Now, in terms of your question about Tmdls, we have worked in Michigan. We have collaborated with what used to be the Environmental Quality Agency. It's now the Environment, Great Lakes and Energy Agency, to help with setting nutrient standards, and we've used some of their data, which includes for Tmdl lakes in our research. But we haven't really gone beyond Michigan. And of course, those are done state by state. So that's really interesting to think about how we might be able to help with managing for nutrients in that way. Thanks for that question.
Next one is from Hayani Setubal. Thanks for the interesting talk, Kendra. I would like to know if you intend to include information about temporary lakes in the database, given the difficulty of mapping these environments that dry out at different frequencies and intensities.
Yeah, that's a great question. We were really excited to see that map that I showed you of the fluctuating connectivity of lakes and how variable, how dynamic that connectivity is, especially in the middle of the US. And so we do have, all of the data that we include comes from very broad, so like national scale databases. And so we start with the national coverage of streams and rivers. And in that, they do list whether something is permanent or ephemeral. And so that's part of where that map came from. In terms of the lakes that are ephemeral, the only way we'd be able to tell that really very carefully, so we have the proxy of whether the incoming and outcoming streams or rivers are ephemeral. The only other way to really get at that would be to use, at least at a broad scale, would be to use remote sensing and look at water levels through time. And I know, so there are some people working on that elsewhere, not in our project, and we're definitely interested in seeing what they come up with and whether we could incorporate that into our research. Because yeah, it's a really interesting idea to be thinking about lakes that disappeared altogether and then come back. Now, some of those are going to be pretty small, but not all of them for sure.
Yeah, definitely. In Brazil, we have entire drinking water reservoirs that disappear for months. The next question is from Nigel. Hi, Kendra. I'm sure there's a lovely socio-ecological paper waiting to be written comparing national customs in lake naming.
That would be so fun. Let me know if you want to do that.
And Pedro Perez Neto is asking, saying, thanks, Kendra, and asking how well can we predict lake features based on spatial modeling, and whether future lakes should be selected on the basis of being not easy to predict and introduce new information.
Yeah, that's really exciting. So one of the things that we're working on right now is taking data from lakes that we, in situ data that we have, and then trying to understand and extrapolate and predict about lakes for which we have no data. And so doing some of that spatial and or temporal modeling. And it is really interesting to think about. We've been talking about novelty, right? So when you try to predict or extrapolate, and you find that there are certain ecosystems or ecosystem types or areas of the country that are very difficult to predict, what does that mean? Are they, how might we think about those as maybe those are unique systems that need to be studied more, protected more, that kind of thing? Or maybe those are, they could be, for example, hotspots for rare species, perhaps. And so we are definitely looking into that and thinking about what does it mean when we have areas that are novel and perhaps difficult to predict.
The next question is from Rosenberg Menezes. It says, "Hi, Kendra. Thanks for the nice talk. In connection with Mariana's question, would these differences in Tp chlorophyll slopes be related to the freshwater acidification that took place in the US in the 70s and 80s?
Yeah, it certainly could be. We've recovered a great deal from that time, but certainly we could be seeing some of those patterns. And that's one of the things I think that's so exciting about going from Lagos Northeast, the Northeastern and Midwestern US, to conterminous US, is all of a sudden we're going to get much larger, wider gradients and different areas of the country that were not affected, for example, by those same patterns of atmospheric movement of those contaminants. And so I think that is a possibility that's part of what's going on there for sure in the northern tier. And it will just be really interesting to be able to look at the whole conterminous US because of the differences in gradients and our ability to look at different types of stressors that happen in different parts of the country.
And then there's another question by Laura Sanchez. "Thanks, Kendra, for the talk. I would like to know if you explore possible changes in Tp and or chlorophyll in these shallow lakes through time."
Hi, Laura. Great to hear from you. We have not yet looked at the shallow lakes through time. Definitely it's on our sort of hopeful list of what we'd like to do in the future. One of the things we've talked about is trying to see if we can detect regime shifts in these lakes through time, for example, and shifting between the clear and turbid states. So we would definitely like to do that. Now, certainly the 2,500 shallow lakes that I was able to show you data from in the talk, when we start to want to look at trends through time, the sample size, of course, goes way down because we have to have, that means we're now needing repeated samples and hopefully at least for 15 years. But for the subset of lakes where we have those, that is definitely something we want to do in the future.
Okay, so you might think that there's no more questions, but you're wrong because I've been trying to type my own questions, but it didn't let me. And so I was curious about two things. One, I wanted to ask if it's possible to make some kind of a risk assessment, either for like a region specific or a lake specific, because you said every region, depending on the type of lake, the type of the soil, watershed use, has its own set of kind of filters. So would it be possible to kind of make like a risk map for global warming, a risk map for fire, a risk map for land use change, something like that?
Yeah, definitely. One of our postdocs, Ian McCullough, has been doing a lot of work with the Lagos data and looking at climate sensitive areas. And so thinking about where in that region, where in the country are going to be most sensitive lakes to climate change. And he's also the one who's been studying the effects of fire and the changing patterns of fire through time. And so, yeah, I think it would be great to come up with a series of sort of risk assessment maps and then try to figure out where is the intersection, right? And so where are those lakes that are both extremely sensitive to climate change, but also sensitive to fire and to invasive species movements, those types of things, I think that would be great.
And my final question is, how would citizen science work in less populated areas? Like, I'm thinking for Brazil, there's so many lakes, shallow lakes, and most of them there's nobody around. So what would you suggest in that case?
Yeah, you know, citizen science works. A lot of what happens with citizen science is it's people who go to a specific lake all the time. They either live on or near the lake, or they go there for fishing or other recreation. And so it's, you know, there isn't as much citizen science involved when you go out into areas that just aren't visited. That's very true. And so I think that's more, those types of systems are ones where we're going to need to figure out, you know, remote sensing capabilities, keep working with remote sensing to try to get a sense of what's happening in those systems, and keep, I mean, we're going to have to keep doing our own work as scientists as well, because it is harder. We could think about, though, you know, if those remote lakes, if it's anything like here in the US, right, the lakes that are in protected areas, for example, that are part of national park systems, that kind of thing, even though people don't live around them, if those are destinations for holidays, you could think about there still being some sort of citizen science programs that happen during those peak visit times, where you could, you know, have an app, you could have a competition for, you know, submit your whatever observations about whatever for those lakes, if they're visited. But when they're out in the middle of nowhere and not visited much, it definitely is harder for citizen science.
Yeah. Okay, so we got one more question. Hi, by Maria Boveri. And she says, hi, Kendra, are you in some kind inspired in the works of Robert Peters, Lars Håkansson, and the empirical team of the 80s and 90s?
Yeah, of course. You know, taking a landscape perspective, it's not like this came out of nowhere, right? There's all sorts of comparative studies and history of limnology that we have built on, and that we, you know, and that's why one of the lessons learned, right, is not just that we need to take a landscape perspective, but that we also need to continue to study individual and small groups of lakes, and to do the more traditional limnology as well, because you can't have one without the other, right? And so definitely building on that empirical work is super important.
Okay, so I think that does it for the questions for your plenary, Kendra. Thank you very much once again. It was a great talk, and also for answering all the questions. So now is normally when we would invite you all to go take
Ali, just a moment before people go out. Hold on. I will put in the chat here for you the links of the Zoom rooms, just in case you don't find them. But before this, I would like to thank you, Kendra, very much for this wonderful talk. It was perfect to introduce our conference. And thank you, Ali, for presenting Kendra to us. We are a little bit ahead of our schedule, and we'll very soon take a break, so you can stretch your legs, go to the bathroom, eat something, get another mug of coffee, and fill your glass with water. But before we leave for the break, I would like to show you a short video about Natal and the Rio Grande do Norte state, in case you want to know, even if virtually, a little bit of our natural and cultural attractions. The oral sections will start promptly at 10:30 a.m. Brazil time, so in 20 minutes, 22 minutes in my clock, on the links that I'm going to send you through the chat to facilitate your access. After the video, then, we will close this webinar, and we want to thank you all for your presence here today. Tomorrow, we will have two very interesting lectures by Matthew Label and Pedro Pérez Neto. Don't miss them. I will send you the links. I hope you get the links here. I'll share you the screen.